So, dear students, in last lecture we mentioned some of the differences between the amylose and amylopectin, and we are understanding the starch. So, as far as the starch is concerned, the starch is made up of the water-soluble fragment, which is called amylose. And in last lecture, I told you about the water-soluble fragment, which contains 15 to 20% of this is called as amylose, and amylose is a long unbranched chain with 200 to 1000 units of the alpha-D-glucopyranose linked together by means of alpha-1,4 glycosidic linkage. So here I just we have discussed the structure now. So the linear polysaccharide, we can say the amylose, which is the water-soluble fragment. Here, two carbohydrate units or two glucose units are linked together by means of alpha-1,4 linkage. So the two alpha-D-glucose molecules, they join, and the glycosidic bond which is formed, the glycosidic bond which is here, it is formed between carbon number 1 of this carbon number 1 and carbon number 4 of the second sugar, and similar things are repeated many times. And as far as the amylose contains 200 to 1000 such units, 200 to 1000 units. Whereas the another part, which is the water insoluble part, which is known as amylopectin, is a branched chain with a having branched chains and having the two kinds of the bonds. So the linear glycosidic 1,4 bonds, alpha-1,4 glycosidic linkage is present, whereas at branch points the alpha-1,6 linkage occurs, made up of the few thousand glucose units, and 20 to 30 glucose units per branch are present. So here I would like to say that this is between the bond between 1 and 4 amylopectin, 1 and 4, and then so between the two glucose molecule units, between the two branching points, you see this is the branching point of this is a carbon number 1 H, and here it leads to formation of a bond with the 1,6 formation of bond with 1,6 linkage. Sorry, leads to formation of bond like this. The new glucose molecules here. This is H and OH, so this will subsequently form the bond with another carbohydrate and the glucose unit. But the branching point, which is here, will lead to formation of a bond here, and this bond is created between the carbon number 6 of this sugar CH2, and this is linked by O, and this type of the bond is known as alpha-1,6 linkage, alpha-1,6 branch side branch point, and rest of the bonds are again alpha-1,4 bonds. And here you can say this is H, OH, OH, H, and this is again H, and here is again linked to new linear chain. So this is alpha D glucose. It's a alpha-1,4 linkage. Alpha-1,4 linkage, glycosidic linkage is here. It's H here, and this is H here. So this is alpha-1,4 glycosidic linkage. This is alpha D glucose. So in this way, on the branch points, alpha-1,6 linkage, alpha-1,4 linkage is there, is repeated, whereas these are the linear bonds. This is the linear bond between the two glucose molecules, and this bond will be here called as alpha 1,6, and this bond is known as alpha 1,6 linkage. So this is the structure of the amylose and amylopectin. So amylose is normally a soluble part of the starch, whereas insoluble part of the starch is known as. So the difference between amylose is actually is not branched, whereas amylopectin is a branched, and amylose is having 201,000 units, and all of them are linked together by means of alpha 1,4 bonds. And whereas in case of the amylopectin, is an insoluble part, and it is having two kinds of the bonds, linear bonds, alpha 1,4, and at the branch points there are alpha 1,6. So this is about the structure of amylopectin. Now we'll take up and the structure of glycogen. So glycogen is also called animal starch. It is known as animal it's called animal starch. So this is a reserve food material, glycogen. It is a reserve food material in animals. So in animals, it is a reserve food material and often called as animal starch. So it is present in high concentration in liver. It's present in high concentration in liver, followed by muscle and brain. Followed by muscles and brain. So this is also found in this. Glycogen is also found in yeast and fungi. So glycogen is also found in fungi and yeast, which are actually achlorophyllous. They also store the uh, carbohydrates in the form of the polysaccharide known as the glycogen. The structure of the glycogen is now, let's look at the structure of glycogen is same as that of structure of glycogen is same as that of amylopectin. Part of starch. Structure of this structure of the glycogen is same as that of the amylopectin part of starch. So same as that of amylopectin part of this is, so how it looks like. The molecular weight of the glycogen is molecular weight is equal to 1 into 10 raised power 8 and the number of the glucose units, are 25,000, or 20. So molecular weight is 1 to 10 power 8, and the number of the Daltons and the number of the glucose units are 25,000. So again, the structure will be same as we have already discussed it above. That structure of the amylopectin part of this glycogen will be same as the amylopectin. That is showing the two kinds of the bonds. It's a branched chain carbohydrate and it's having alpha 1,4 bonds and alpha 1,6 bonds at the branching points. And the glycogen is very, very important for animals. It says used as a reserve food material in case of the animals, and is utilized at the time of the need. Clear? So this is all about this glycogen. So you must remember this, and same is that of the amylopectin, means that there are alpha 1,4 bonds and alpha 1,6 at branching points. Alpha 1,6 at branch points. So this is all about it. Structure of a polysaccharide found in plants known as cellulose. Cellulose is again a homopolysaccharide and occurs predominantly in the in plants as the most abundant organic substance. It's found in the plant cell walls Cellulose is absent in animals. So this homopolysaccharide cellulose is not found in the animals. However, there is a group of animals which are called Urochordata. In Urochordata or in Urochordates, test or tunic of body is covered by a carbohydrate tunicin which is allied to cellulose. So cellulose is composed of the beta D-glucose units. Beta D-glucose units. So everywhere in cellulose, the carbohydrate units are glucose, but the glucose is a beta D-glucopyranose units, which are linked together by means of the beta-1,4 glycosidic bonds. So just like you can understand, the glucose units will be beta glucose units like this. So another glucose unit will be here. Again a beta glucose. So this is the sign, this is the beta glucose. This is D-glucose, but this is the beta hydroxyl, and this is the beta hydroxyl in this. And here we know and here this glucose and this is linked together. So this is beta carbon number 1, 2, 3, 4, 5, and 6. 1, 2, 3, 4, 5, and 6, and this is the beta 1,4 bond in cellulose. And here it will be N times. And this is the beta D-glucopyranose. And it's another unit of beta D-glucopyranose. And this is the structure of the glucose. And the final disaccharide which is obtained from the glucose will be like same like this. That will be C12H2211, and that's called a cellobiose. Cellobiose. Cellobiose is a beta 1,4, or we can say it's a beta D-glucoside, or beta D made up of the two glucose units which are beta D-glucopyranose units. Cellulose is disaccharide of cellulose origin, uh, with the formula C12H22O11, and this having the same structure like this, and n times this structure is here called as cellulose. And as per the cellulose, it's not digested in human bodies. In humans, cellulose is not digested because in human digestive system, they're all enzymes can digest starches, which are alpha amylases. Alpha amylases. Since we know alpha amylase can bring about the breakdown of the alpha 1,4 bond, and in humans there is no alpha beta 1,4 beta amylases, so the cellulose remains undigested and acts as a and is used as a dietary fiber. It's very, very significant because acting as a dietary fiber, whereas in some animals the cellulose is digested. Cellulose is digested in ruminants. Is digested in ruminants and herbivorous animals, which contain the microorganisms in their gut. Cellulose is digested in ruminants and other herbivorous animals. Because their guts contains bacteria present in their gut can produce cellulase. In their gut can produce cellulase that brings about the digestion of the cellulose. So in humans,